also. So this outline essentially covers the BAB studies, methods to determine the BAB, and various challenges related to the each therapeutic area, and also a typical general challenges related to BAB studies, and also how to deal with the product failures, and some conclusions. BAB studies are essential part of the ANDA submission to Europe or to US regulatory or any other country. It represents the therapeutic equivalence, means if you determine the concentration or blood levels or the PK of a compound or so then it represents the, it's almost indirectly related to therapeutic equivalence and contributes to the success of generic pharmaceutical industry. Because without which study, there is nothing can replace these studies. These are the methods to determine the BAB studies. It is a general for general audience who are novice audience. There are four types of BAB determination. One of them is pharmacokinetic studies. It's just measuring the blood concentrations. It's a very rational approach, and most of the oral solids are being done through this approach only. It's a very valid, scientifically valid approach, typically conducted in healthy volunteers, and you need to determine the BE through statistical evaluation so that the study is meeting the requirements. Then comes the pharmacodynamic studies, generally not recommended for solid orals. A valid biomarker is required. It means you need to validate that marker. And then comes the comparative clinical studies, recommended when PK and PD approaches are not feasible. Means there is no valid biomarker or you cannot determine the concentrations in the blood, then you go for the clinical endpoint studies. Most of the like local acting medicines and some ophthalmics and some other therapeutic areas it is being used. And in vitro studies, it is recommended for BCS class 1 drugs, and still BCS class 1 means you need to conduct some studies to prove that it is BCS 1. Literature support does not guarantee, you need to conduct the studies. Then the industry, how worth it is industry really using this approach? I think it may be known to the pharmaceutical industry. So these are the some of the categories of therapeutic categories where some challenges are being brought in. Typically, it is local acting products, topical products, ophthalmics and respiratory products, where nasal space and inhalations are there. Liposomal injectable products and complex drug products like biosimilars. There are other categories are there which I didn't note here. Coming to the local acting GA products, where the absorption is very limited and it is not absorbed into the system. It's not possible to quantitate the concentrations. You cannot measure reliably. And definitely it invites go for this clinical endpoint studies like sucralfate suspension, where they it is recommended to conduct a clinical endpoint study with ulcerative colitis patients. And in vitro studies are being done for some of the local acting products, where like cholesterol. Its bile binding capacity is being measured in vitro and it is proved as a BE criteria. And the PK studies, for some it is being explored, and it is the FDA is also okay with this type of molecules or products. Example is misoprostol and sulfasalazine. When you want to conduct local acting GA product studies, I think these are the 4 factors we need to look at when you want to decide or when you want to talk with FDA what is the best approach to go for a BE study. It's because of this. Absorption varies from site to site. Clinical relevance is still questionable. That means whether if you measure the concentrations across the stratum corneum or whether it is relevant or not, it is still a questionable one. And different labs, different results from different labs are observed. So the interlab variations are very high. So coming out with some kind of a rational approach is a difficult one, and it is a challenge for the pharmaceutical industry. Coming to the ophthalmic products, I think some of the companies have started doing these ophthalmic products. where most of the time it may be like Q1, Q2s are okay with it, where you can show the quality, quantitative and qualitative composition, similarities and those kinds of things. Coming to the suspension emulsions products, loteprednol and loteprednol plus tobramycin ophthalmic suspension. So very difficult to conduct study. Large number of volunteers are required. You need to collect the aqueous humor to go into the design. So it is very restricted from the FDA.
Thank you. Product is conventional dosage form. The same drug like to formulate as sustained release dosage form. So the same bioequivalence study can be conducted.
Yeah, it all depends on the where you are in your development cycle, where you want it. If there is no sustained release dosage form in the market.
Yes, sir. If the such type of sustained release dosage form,
Then you have to conduct against the IR formulation only.
And if I want to go for combination of two drug products, which type of combination presently it is not available in the market, but individually two drugs are available, two innovator products are available. But such combination, if it is not available, then sir?
Yeah, basically it depends on the regulations. If it is a combination product, you have to do a limited clinical trials. It depends on the regulatory. If it is for US, you may have to do a limited clinical trials, as well as the bioequivalence studies, individually and combination also.
So it will not come under NDA. It will come under only NDA.
It comes under paper NDA. Because typically you are going to use both studies information, means individual different product clinical information you are going to use and submit it. You are going to cite that information to FDA, and you are using the additional data also.
If I change the dosage form also, it's the same thing?
Yeah, dosage form change or salt or any ester or anything you are making, you need to, it's like a FDA, it is a must to that, you have to go for a paper NDA, and for European it's not required, you can directly submit it.
In India, sir?
In India also not required.
Thank you, sir.
Thank you.
Maybe 2 to 3 years, we have realized that it has got one disadvantage or one major side effect. Like after long taking for a lot amount of time, then you realize it. But the drug is of only type in its use. Like there is no other substitute or no other competition, and we've invested millions and billions of dollars in it. Then what are we supposed to do? Like what should be our next plan? Should we go for another R&D, spending billions, or try to modify a bit, or too late?
See, you are talking about something like orphan drug, where there is no established treatment available. So in those cases, I think if it is a very rarely occurring adverse event, say 1 in 10,000 or 1 in 1 lakh or something, then it won't be a much issue. I think the regulatory will allow it, will allow to continue, because the benefit is more than the risk you are involved in that. So it will be okay with it. It all depends on the regulatory country.
So then our next step will be like kind of...
So if there are so many number of people are affected within no time, then nobody will allow for it. If something is there like very rare adverse event, it's happening in say, maybe the population, the patient population itself will be very limited, maybe say 200,000 people only. So in that case, I think 1 in 10,000 people is affecting, not an issue. Regulator also won't object it, because you are making the other 200,000 people survival.
Thank you.
Thanks.